Okay, we can go ahead and get started now and make sure I got my sound up. And today we're going to start on the muscles from the physiology side. And just a reminder that registration opened yesterday, and if you need to have the registration hold from meeting with an advisor removed, just let me know, send me your W, and I'll go ahead and do that for you. And it's no big deal. Reminder that if you're going to take AP2 in the spring with me, and this is not a plug, but if you're going to take it with me, the hybrid class is going to be offered both on ground and on Zoom. So you'll have the option of attending. You get it's a pretty good deal. You get to choose every day whether you're going to come to class or you're going to Zoom. So there's no difference. You're going to get the same lecture virtually that anybody that comes on the ground will get. So if you don't live in the Sevierville area, you can still take the class like we're doing right now, a virtual class. If you live in the Sevierville area and you want to come to class and get the hands-on lab, for example, and see any demonstrations and lecture, you can come to class. You can decide what to do every day. So there's a tremendous amount of flexibility here in the hybrid class in the spring, where this is sort of an experiment, see if it works. And if it does, we can do other classes this way too. So anyway, so if you want to register for AP2, that's why it's going to be both on ground and Zoom at the same time. And of course, the lectures will all be recorded and posted so you can watch them later on too. I hope I didn't make that too confusing. So anyway, lab this afternoon, we're going to start taking a look at the nervous system. I know we haven't got there yet. We're one chapter away from the nervous system, but this will be a good introduction to it. So anyway, any questions on anything? Okay, and I'm going to go ahead and start with what we call chapter 9 on the muscles. Go from there. And you should all see the slide that says Human Muscle Physiology, Chapter 9. And now you should see a cute little mouse. Okay. Now this is not one of my pets, but the reason that we have a mouse here is this is where the name muscle comes from. Early anatomists thought that the movement of a mouse underneath a blanket, apparently they had a lot of problems with rodents in those days, and a mouse running underneath the blanket looked just like our arm muscles did. Well, they didn't call them muscles. They didn't know what they were coming up with a name for it. And so the bulge underneath the blanket that the mouse made when it ran through there looked just like the way the skin bulged when you flexed your arm or your leg. So the term for a mouse, the Latin term for a mouse is mus musculus. And so they started calling them musculus or muscles for the structures that we now know as muscles. So that's where it's related to the mouse. Anyway, little piece of physiology trivia. Who says we're boring anyway? Okay, muscle tissue makes up about half of our body mass. Most of it is skeletal muscle, or what we call voluntary muscle or striated muscle, the muscle that we can control. Most muscle is under our control, but there are some significant exceptions. Muscle cells of any kind take ATP and turn it into mechanical energy. Now, all a muscle can do is contract. That's all it can do. It can contract and then it relaxes. It contracts and then it relaxes. That's all it can do. Now, we have three distinct types of muscle tissue in our bodies. They all exhibit the same characteristics, and there are a variety of functions they do in addition to contracting. So, terms. The prefix myo, or mis, or sarco, all refer to muscle cells. Sarcoplasm is the cytoplasm of a muscle cell. Sarcolemma is the cell membrane of a muscle cell. Myofibril is a structure inside the muscle cell, and myofilaments are the contractile proteins that make up the myofibril. So you're going to see all these terms here, and I will confess there is a certain level of confusion. Maybe I should learn to speak correctly. A certain level of confusion in these terms because they're similar terms and some are used interchangeably. So, now, the muscle cell types, there's only three. Skeletal muscle, which is the bulk of our muscles. That's what we call voluntary muscle or striated muscle. It's what we've been looking at in labs, like the biceps and the triceps and the sternocleidomastoid. It's the muscles that we have direct control over. We tell our arm we raise our right arm. We say, okay, right arm, go up. Not quite like that, but that's voluntary control. And put your arm back down. So we put our arm back down. That's voluntary control. That's what we mean by under control. We tell our skeletal muscles what to do. Cardiac muscle, only in the heart, cleverly called cardiac. Cardiac muscle is involuntary. It's also striated. That's its only similarity to skeletal muscle. It's striated, but it's involuntary, and it is interconnected to all the other cells in the heart. Every cell in the heart is connected to every other cell through gap junctions, and those gap junctions allow the heart to contract as one big cell. Because when the heart contracts, every cell contracts together. In smooth muscle is found totally different than the other two. It's also involuntary. Smooth muscle is around the digestive system, around blood vessels, around our hollow organs like our urinary bladder. We don't have any control over it. It's what will propel food through our digestive tract from the mouth to the anus. It's what will enlarge or shrink our blood vessels as needed to move blood around. It will open up our airway or close our airway. So we don't think about it. We don't have to. It's all controlled by the central nervous system. And the smooth muscle, not striated at all, totally different in appearance than the other two. Skeletal smooth muscle is pointy. It's like a spindle. So, and we call the cells muscle fibers. So we use the term muscle fiber interchangeably with skeletal muscle and smooth muscle. In particular, in skeletal muscle, when you have a discrete organ like the biceps or the gastrocnemius, the muscle fibers, the muscle cells, they're called fibers because they're long and stretched out. The muscle cells are called fibers, and they run the length of the muscle. And you'll have thousands and thousands of muscle cells in a muscle, but every one of those muscle cells will run, however long the organ is, the biceps, the triceps, the gastrocnemius, any of the quads or hamstrings, whatever muscle we're looking at, the cells will be as long as the muscle is. They run the entire length of the muscle. There we go. Okay, so skeletal muscle organized into discrete muscles. When looking at those again, you know, the biceps and the triceps and the brachioradialis and things like that. They attach to bone. They attach to their underlying skin. They have an origin and an insertion. The muscle fibers are the longest of all muscle cells. They're the largest of all muscle cells. And they also have striations. That's what gives them their striped appearance. You know, what looks like alternating light and dark bands. Call it voluntary muscle, call it skeletal muscle, call it striated muscle. It's all the same type. Skeletal muscle is very powerful. It has the ability to exert great force. It can contract rapidly, but it will get tired out quickly. You can do powerful contractions with skeletal muscle, but you can't keep it up indefinitely. Whereas smooth muscle can contract and move food through your digestive system constantly. Your heart can beat every 0.7 of a second for 100 years, but skeletal muscle, while very powerful, will get tired easy. So again, for voluntary muscle, skeletal muscle, striated muscle, all the same thing. The organs, the discrete organs that we call the muscles, that we're looking at in lab. So, cardiac muscle, only found in the heart, makes up not all of the heart, but the bulk of the heart. Cardiac muscle wraps around the heart in a series of bands, so it has something to contract against. It is striated. That's its only similarity to skeletal muscle. It is involuntary. The cardiac muscle branches. The cells are thicker, and they branch. They branch at what we call the intercalated disc. The disc is where they're connected to other cells with desmosomes and gap junctions. Desmosomes give them great strength and flexibility. Gap junctions allow nerve impulses within the heart to travel from cell to cell very quickly, because our heart will contract every 0.7 of a second. That's fairly quick. And every cell inside the heart contracts together at the exact same time. It isn't like skeletal muscle. In skeletal muscle, we use the muscle cells we need to do something. The amount of force it takes to turn a doorknob is different than the amount of force you would use to push a 100-pound weight or lift a 100-pound weight. You apply different forces to different situations based on how many muscle cells you're going to use. In the heart, it's totally different. In the heart, you use every cell all the time. Smooth muscle around the hollow organs like the stomach and the gallbladder and the urinary bladder, around our airways, for example, around all of our blood vessels to control the movement of blood. We don't control smooth muscle either. We don't control cardiac muscle. We don't control smooth muscle. It's not striated. It's involuntary muscle. In terms that, remember, it's not striated, it's visceral. Never really used that term for it. Usually smooth muscle or involuntary are the key terms for smooth muscle. So, yeah. Examples of what they look like: striated muscle, skeletal muscle, lots of nuclei. All these little purple chunky things are a nucleus. Skeletal muscle is what we call multinucleated, because when we lay those muscle cells down in utero, when we're developing in the uterus, we form skeletal muscle cells out of numerous smaller cells. So, and we retain the nucleus. And since we can't reproduce muscle cells, skeletal muscle cells, it's no big deal. The cardiac muscle, striated, similar to skeletal muscle, one nucleus per cell. They branch. You can see how they branch apart, how the lines stretch out. And they have these lines here called the intercalated disc where the cells collect. Over here is the spindle-shaped smooth muscle, one nucleus, no striations. And we're going to talk mostly about skeletal muscle in here. And whether it's cardiac muscle, skeletal muscle, or smooth muscle, however, there are four things that all muscle cells have in common. They are excitable. They will respond to a stimulus, a nerve impulse, whether generated by itself, like in the heart, or received from the nervous system itself, like in skeletal muscle or smooth muscle. The muscle will respond. It will respond to the stimulus. So if it's excitable, it responds. If you get a stimulus, it will respond. And their response is the ability to contract. They will contract and shorten when stimulated. That's all a muscle can do. And all the cells can do is contract. They contract and then they relax. They contract and then they relax. That's all they can do is contract. Muscle cells can contract. They can be stretched. When we exercise, we make our muscles work harder. We stretch them. We push them to their limits and we stretch them, and they get stronger. And then they return after they're stretched. They will rebound to their original position. So muscle cells are excitable, contractible, extendable, and elastic. So, okay. What do they do for us? Well, there's four things they do. They allow us to move. The number one thing that muscles do is allow movement. Our heart pumps blood. That's moving blood through our bodies. The smooth muscle squeezes food through our digestive system from the mouth to the anus. We walk and talk and stand upright and sit down. We make words and sounds. We eat food. We manipulate our environment, as they say. So all these things are movement that our muscles allow us to do, whether it's smooth muscle or skeletal muscle or cardiac muscle. We also use our muscles to hold our body in the right position, whether we're sitting upright, whether we're standing upright. We maintain our posture. We maintain our position. We don't lean against the wall and slide down onto the floor, because we maintain our posture. We stabilize the joints. Muscles pull on bones and hold our joints together. Muscles pull on bones, hold our joints together, particularly at the pectoral girdle and the pelvic girdle. Pectoral girdle in particular, because there's not a lot of bone structure there. Well, there's plenty of bone structure, but like the glenoid cavity is not a very good socket. It's just more of a flat surface than anything else. But our muscles, particularly like the rotator cuff muscles, hold the humerus against the glenoid cavity very nicely, very tightly. And that gives it its powerful movement, but also flexibility. And something else we see is that muscles generate a lot of heat. 60% of the energy that's in a molecule of glucose is released as heat. We only capture 40% of it, since most of the cellular respiration is taking place, since a pretty good amount of cellular respiration is taking place in our muscles, 40% of the energy in a molecule of glucose goes off to do stuff by making ATP. The other 60% of that energy in a molecule of glucose is released as heat. And one of the side effects of that is that that's how we maintain a constant body temperature. So, while yes, we do lose the heat, the energy is not destroyed, it just is dissipated. Heat energy just spreads out. But our bodies are designed to run at 98.6, 37 degrees Celsius, and everything about the chemical reactions in our body are based on that temperature. So let's take a closer look at skeletal muscle. That's what we're going to focus on primarily here anyway. Skeletal muscles are organs. The biceps is an organ. The triceps is an organ. They have a blood supply. They have a nervous system. They're surrounded by connective tissue. And they have attachments, origins, and insertions. So, it meets the definition of an organ because it has multiple tissues here. It has connective tissue, it has epithelial cells, it has muscle tissue, it has endothelial cells, it has nervous tissue, all coming together in these discrete organs. We have a very sophisticated control system here. Every muscle is supplied by a nerve, an artery, and veins. Blood going in, blood going out. We have control over every muscle cell in a muscle. We have nervous system control. A nerve is in contact with every cell. There's a connection to every cell with a nerve. So we can control a muscle down to the level of the individual muscle cells. So we have this remarkable control system in place here. Now, when a muscle cell is stimulated, it will contract to its fullest extent, meaning that there's no sort of partial contraction of a muscle cell. It either contracts fully or it doesn't. When it's stimulated, it contracts fully. When a muscle cell receives a nerve impulse, it will contract as powerfully as it can, a full contraction. Now, where we have even greater control, though, is how many muscle cells in the muscle are we going to contract? We can't tell the cell to only contract a little bit. We tell the cell to contract fully. How many cells do we need to use to get something done? Are you going to use more muscle cells to throw a ping pong ball or a baseball? Well, obviously, you're probably going to use more muscle cells in your arm to throw a baseball than to throw a ping pong ball. The force that we apply to do things is based on how many muscle cells we're going to use. Every cell that's stimulated will contract to its fullest extent. It will have a powerful contraction. Each cell will have a powerful contraction. How many cells do you need to use? In doing this, there are tremendous demands for oxygen and for glucose. Now, skeletal muscle stores oxygen in myoglobin. Myoglobin is the red pigment. We call it a respiratory pigment. It stores oxygen in muscle cells. It's what makes meat red. Beef looks red. You can see beef at the meat counter in the meat section at Food City, for example. That beef is red because of the presence of myoglobin. Myoglobin turns red in the presence of oxygen, and it's holding oxygen. What we claim is blood dripping out of raw meat. You would buy a packet, buy a steak or ground beef or a roast or something at Food City or Walmart or wherever, that's not blood, that's myoglobin dripping out. Beef, the sides of beef usually are drained of blood long before it ever gets sliced up. Anyway, so we have lots of stored oxygen in the myoglobin. We have lots of stored glucose in glycogen within the muscle. There is so much oxygen and glucose present that skeletal muscle cells will usually not die for about 8 to 12 hours after death. Individual muscle cells will often stay alive for hours after death. In fact, cardiac muscle cells, even if the blood supply to the cardiac muscle cell is cut off, it will stay alive for an hour on its stored oxygen and stored glucose. Stay alive for an hour. And so when dealing with a heart attack, for example, and a heart attack is caused by a blockage in the coronary artery that shuts off blood supply to cardiac muscle cells downstream of the blockage, you've got an hour to fix that blockage before the damage becomes permanent. Okay. Now, something else we notice is that muscle cells don't attach directly to bone. Yes, we know there's an origin of insertion where the muscle pulls on a bone. That's how muscles work. They amplify their strength by using bones as levers, but they don't attach directly. They attach indirectly. Each muscle cell is surrounded by a layer of connective tissue. At the bottom of the list here, the endomysium is connective tissue. It's areolar connective tissue, that stuff you see in the mesh type. It is connective tissue that surrounds the individual muscle cell or muscle fiber, same term. Same fiber and cell mean the same thing. So you have this endomysium that surrounds every cell. It's its outer layer of connective tissue. When the muscle cell contracts, it pulls on the endomysium. Now the cells are clustered together within the muscle in groups, and those groups are called fascicles. So you'll have a couple hundred muscle cells in a fascicle. And so the muscle, if you were to do a cross section of it, you would see all these clusters here of fascicles, and the cells would be in the clusters. Now the fascicles, the cluster of cells, is surrounded by another layer of connective tissue called the perimysium. The perimysium is this intermediate layer of connective tissue. Now the individual cells, endomysiums, are continuous, meaning they're in contact with the perimysium. So if a cell contracts and pulls on its endomysium connective tissue covering, that endomysium will be pulling on the perimysium. Now, one cell is not going to, the force of one cell, while very powerful unto itself, is probably not enough to cause even a ripple in a perimysium, but it could. Now, all the fascicles together, each one of those surrounded by a perimysium, each perimysium that surrounds the fascicle is connected, or as we say, is continuous with the outer covering of the muscle called the epimysium. So the perimysium surrounds the fascicle and it's continuous with the epimysium. So when a fascicle contracts, it pulls on the epimysium, the outer covering of the muscle itself. One muscle cell pulls on the endomysium, which pulls on the perimysium, which pulls on the epimysium. One muscle cell probably is not even noticeable, but 1,000 muscle cells is, maybe even 100 muscle cells. Looks something like this. This is a cross section up here in the corner of a section of muscle. And these areas outlined by white are the fascicles. This is a fascicle here. Each fascicle is surrounded by the paramycium. The individual cells, like we see down here, this is a cell. Here's in this illustration, in the artwork, that's a fascicle sticking out from the cell, from the muscle. This is a fascicle. There we go. This is a cell coming out of the muscle of the fascicle. This is a cell, and all these other structures here are cells. So maybe there's 50 cells in a fascicle here. Each cell or muscle fiber, same thing, is surrounded by the endomysium. All of these cells collectively in the fascicle are surrounded by a paramycium. The layer of the endomysium connects to the paramycium. The muscle is made up of clusters of all these fascicles. Each paramycium is connected to the epimysium, and the epimysium then forms the tendon made out of collagen, which pulls on the bone. So the muscle doesn't pull directly on the bone. It pulls on connective tissue, which pulls on the tendon, which pulls on the bone. It protects the muscle cells from being damaged when they're contracting. We know this already from lab. Every muscle has an origin and an insertion. You can have multiple origins. You can have 2, 3 connections. The sternocleidomastoid has it attaches at both the sternum and the clavicle. Its insertion, the movable bone, is the mastoid process. So the insertion attaches to the bone that moves. In this case, when the head turns, the mastoid process is moving. But the sternum and clavicle don't move. So how do we get muscles to contract? Well, we need to take a little deeper look here. The outer covering of a muscle cell, the cell membrane, is called the sarcolemma. Underneath that, we have the cytoplasm called the sarcoplasm. Within the cytoplasm, we have lots of glycogen storage present there to break down glycogen to glucose, and the muscle is filled with myoglobin. Now, within the muscle itself, within the muscle cell, we have something called myofibrils, the sarcoplasmic reticulum, and T-tubules. Myofibrils are rods that run the length of the cell. The myofibrils, if you go back here to this picture right here, all these little round things. So this is a muscle cell right here, where it says muscle fiber. All of these little round structures in here are the myofibrils. These are these rod-like structures that run the length of the cell also. So a myofibril, one cell can have 1 million myofibrils. The myofibrils make up, all the myofibrils together make up about 80% of the mass of a cell. The bulk of the cell is made of myofibrils. The myofibrils give us the striations in here. They form the primary contracting unit of the muscle called the sarcomere. And they are made up of myofilaments. Myofilaments are made up of actin and myosin proteins. You've heard of actin before. Actin is the contractile protein that we use in cytokinesis to pinch the cytoplasm of in half when the daughter cells are forming after mitosis. So striations are found in the myofibrils. The sarcomeres, the contracting unit in every cell, is found in the myofibril. The myofilaments are the contractile proteins that are found in the myofibril, and the myofilaments are the actin and the myosin. So here's a myofibril right here. Better picture than I showed you earlier. Here's a cell. This is a muscle cell right here, or a muscle fiber. We know it's a cell because it has a cell membrane. It has multiple nuclei. Within the sarcoplasm of that cell are thousands of these myofibrils jammed in here like straws or rods that run the length of the cell. And you can see, though, that the striations are very visible. We have alternating bands. We are alternating dark and light bands. Dark, light, dark, light, dark, light. So this is what a muscle cell looks like. Packed full of myofibrils in here, and we have many nuclei, and we have alternating light and dark bands, which give us the appearance of the striations. So the myofibrils are caused by the myofilaments that are present in here. So first of all, what are the bands? You have a dark band called the A band. You need to remember, this is one of the things you need to remember. The dark band is called the A band. The I band is the light band. So you have an I band and an A band. So you have an I band and an A band. You have an M line, which is in the middle of the dark band. And more importantly, you have a Z line, which is in the middle of the light band. So the I band is the light band, and the Z line is in the middle of the I band. So you can see it right here. It's a little line that runs down. Now there's your light band right there. This whole thing is a light band. And the Z line is this little tiny band right through the middle here of the light band. The dark band is the A band. So you have a dark band and a light band. The dark band is the A band. The light band is the I band. There's a Z line in the middle of the light band. Now, to cause these striations, they're formed by the presence of the myofilaments. So the myofibrils appear to be striped thanks to the myofilaments. And this is where the actin protein and the myosin protein come in. The actin myofilaments are called the thin filaments. Actin's a protein. It's a very thin, double helix structured protein. And the actin shows up on either side of the A band. The actin forms the I band and part of the A band. The actin is attached to what we call the Z line. And they call it Z disc. Some of them call it Z disc, but the proper term is the Z line. The myosin are the thick filaments. They're found only in the A band. They're what make the A band dark. Because remember, the A band is the dark band, and the I band is the light band. We have alternating bands, light and dark, light and dark, light and dark. The I band is made up of the thin filaments. The A band is made up of the thick filaments, the myosin. A sarcomere will extend from Z line to Z line. I know this is probably pretty confusing at this point. Let me show you what it looks like. Now this is a schematic of how it appears. Here's our Z line right here. It's just proteins that the actin's attached to. All these little blue lines here are actin. All these thick red structures are the myosin. Now this Z line is a protein that actin's attached to from both sides. Z line, actin, actin, actin. And another Z line over here. The A band is this area that is dark. The A band is this area where we have an overlap of actin and myosin. Right, and here's the A band would be this area where we have the blue lines over the red lines. That's the A band. The I band is the area where we only have actin. So the I band's here and at the other end. This is probably a better illustration here. So here's a Z line right there. There's a Z line, there's a Z line. See this light area on either side of the Z line? That's the I band. The I band is only contains actin. There's another I band there, and the Z line's in the middle. The dark band, the A band, is the overlap of the actin and the myosin itself. So we have the dark band here, light band, dark band, light band, dark band. Now from Z line to Z line on each, on this myofibril, remember these are the myofibrils. And what we're looking at are the actins and myosins are the myofilaments. From Z line to Z line is called the sarcomere. And the sarcomere is the contracting unit of a muscle cell. Contractions of the muscle cell take place in the sarcomeres. So let's take a look at these myofilaments. Myosin, the thick filament, made up of myosin molecules. Each myosin molecule has 2 strands of protein wrapped around each other to form a tail and 2 heads. It looks sort of like a golf club, if you will, the way it's laid out. Each myosin molecule joins about 300 other myosin molecules to make a thick filament. Every thick filament is made up of about 300 myosins, and every myosin has 2 heads. And the heads are arrayed so that if they move, they all move towards the middle of the A band. So if the myosin heads can pivot, but they pivot in the direction of the middle of the A band. So whether they're on the left side of the A band or the right side, when the myosin heads pivot, they pivot towards the middle. The myosin heads have on their very tip something called the actin binding site, which means that they can bind to the actin. And again, they also have a hinge right behind the heads that allow the heads to pivot in one direction. So you'll have about 300 myosin molecules, you'll have about 600 myosin heads in a thick filament. And so there's billions of thick filaments in a muscle cell. In one cell, you're going to have billions of these filaments. The thickness of the filament makes up the bulk of the dark band. The thick filaments make up the bulk of the dark band. Now, overlaying the thick filament is the thin filament, the actin. Actin, it's probably not really blue. And the actin protein doesn't look like blueberries. But the actin molecule, the thin filament is put together in a double helix structure. And it has 2 proteins, 2 controlling proteins woven through it. Now, the actin, each actin protein has a binding site for myosin. So the myosin binding site is where the myosin head will attach. The myosin has a binding site for the actin on its head, and the actin has a myosin binding site on its subunit. They come together at that spot. Now, so we have all these arrayed here. Now you'll notice that there is a protein called tropomyosin woven through the double helix. Tropomyosin lays on top of the binding sites. myosin can't grab onto the actin and can't bind with the actin if the binding site is blocked. And it's blocked right now. The way you see it, the tropomyosin is blocking the binding site. There's another protein there called troponin. Troponin is the key. Troponin controls the position of the tropomyosin. If troponin becomes stimulated or activated, it will roll over and it'll drag the tropomyosin with it and expose the binding sites. The tropomyosin and troponin control the ability of myosin and actin to bind. So without activated troponin, no binding. So these are the myofilaments that make up the myofibril, the actin and the myosin. The actin is the thin filament. The myosin is the thick filament. The thin filament overlays the thick filament. They overlap in the A band. So they overlap in the A band. They make the A band the dark band. Where we only have the actin, we have the I band, the light band. Now, there are some other proteins involved here too in our myofilaments. We have an elastic filament that attaches to the end of every, it connects the actins to the Z line. So at every Z line, you're going to have elastic protein holding on to the actins from both sides, coming together to form at the Z line, so that the actins can rebound after they get stretched, after they contract. So because we certainly want our actins to come back to their original shape. Now, another protein here called dystrophin. And this is a pretty important protein. Dystrophin links the actin to the cell membrane, because it's great that the actin and the myosin will move. I mean, that's wonderful. But unless the cell membrane moves, nothing's going to happen. So we have to have the cell membrane moving at the same time. And we link to the cell membrane with this protein called dystrophin. It holds the sarcomere, the individual contracting unit, together. It keeps it shape, so everything slides together like it should. Now, dystrophin can cause some health issues. There is a condition known as muscular dystrophy. You may have heard of it. Duchenne's muscular dystrophy is the most common type. There are numerous types. So Duchenne's muscular dystrophy, it's caused by a defective dystrophin protein. It's caused by a recessive gene. The recessive gene, it's what we call a sex-linked gene. Thank you. Oh, okay, I see what you're working on. So, okay. So a sex-linked gene shows up, in this case, on the X chromosome from the mother. Duchenne's muscular dystrophy is a sex-linked recessive disease. It's caused by a recessive trait that gets passed from the mother. The mother is the carrier, and the male offspring is the recipient. Now, there are a wide variety of sex-linked disorders. This is one of the very serious ones. Some are a little less serious, like male pattern baldness. Male pattern baldness is a sex-linked disorder. Again, a gift from our mothers, from mothers to their sons. Red-green color blindness, sex-linked disorder, passed from mother to son. Hemophilia, the mother is the carrier, the son is the recipient. So it's nearly always in males because it's very hard to have, in A and P there are very, very, very few absolutes because there are always exceptions. But in these sex-linked disorders, it's nearly always the male. So the mother passes it on to the male on the X chromosome because males have an X and a Y chromosome. The Duchenne's muscular dystrophy, it occurs at a very low rate, 1 out of every 3,600 births. So that's a pretty low rate there. What happens is the recessive gene produces a defective type, directs the production of a defective type of dystrophin. And so it doesn't attach to the sarcolemma well. And the sarcolemma can rip and tear. And when it does, it allows calcium to flood inside into the sarcoplasm and start damaging the cell and maybe eventually triggering apoptosis. You start seeing this early on in life, somewhere between 2 and 7, as the child is developing and learning to walk. It starts to starts to regress in its movements because muscle cells are not controlled anymore like they should be. And eventually it will kill the affected individual, usually from respiratory failure, perhaps in early adulthood, frequently in adolescence. So, and so you see, since it's a genetic disorder, it's hard to treat. It's very difficult. It's difficult to treat in here because of the. It's not like it's not like you can just apply an antibiotic or something here. It doesn't work that way. The gene affects this. Now, there are numerous approaches to trying to treat this with gene therapy, just like for cystic fibrosis. Cystic fibrosis is another inherited disorder, but that one comes from that one is a homozygous recessive gene. You have to have the recessive trait from both parents. Here, it comes just from the mother. Okay. Now, structures. Let's pay attention to how these structures allow a muscle to contract. The sarcoplasmic reticulum is a key structure here. We know what the sarcolemma is. It's the cell membrane. We know what the sarcoplasm is. It's the cytoplasm. The sarcoplasmic reticulum is similar to the endoplasmic reticulum in other cells. It's very similar to smooth ER because it's a series of tubes that surround all of the myofibrils. The thousands of myofibrils in the muscle cell are surrounded. Each one is surrounded by these tubes, the sarcoplasmic reticulum. They run the length of the cell, and they run the length of every myofibril. And these tubes are storage facilities to hold calcium. They're packed full of calcium. The sarcoplasmic reticulum contains high levels of calcium. Now, at the end of each sarcomere, at the location from the Z line to the Z line, what we have here are cisterns, enlargements of the tubules. Now, a cistern is a storage tank, essentially. You would have a cistern like a rainwater cistern. It's like, many people have rain barrels now to collect rainwater to use in their gardens or as a backup water source, for whatever reason. And before rain barrels, and even while some places would have a underground tank that they would fill up with rainwater. That's what a cistern is. Well, we don't bury our terminal cistern underground. Our terminal cisterns appear at the end of each sarcomere as an enlargement of the sarcoplasmic reticulum. The whole thing is filled with calcium. So you got a Z line, you've got a cistern on each end of each side of the Z line, packed with calcium. every myofibril is surrounded by all these tubules, all packed with calcium. Running down through the cell, from the sarcolemma throughout the cell, are a series of tubes or tubules that connect to the surface of the cell and will run down and surround every myofibril. Myofibril at the individual Z lines. These are called the T tubules. What these are nothing more than extensions of the cell membrane. And they run down here, they surround the myofibril at each individual Z line. So there are probably millions of T tubules in every muscle cell. The T tubules allow a nerve impulse that reaches the cell membrane and can then be passed down through the entire cell and take that nerve impulse to the terminal cisterns. So it looks something like this. This is a cell, and each one of these are myofibrils. Each one of these red things here are myofibrils. The blue structures are the sarcoplasmic reticulum that surround every myofibril. These enlargements here on either side of this white line are the terminal cisterns. The white lines are the T tubules. The T tubules extend all the way to the surface of the cell. They are part of the cell membrane. And so they are in contact with every myofibril. The T tubules are in contact with every myofibril at every Z line, here and here and here and here and here, and all the way throughout the entire cell. The whole purpose of the T tubule is to transfer a nerve impulse that starts in the surface of the cell, on the cell membrane, to transfer a nerve impulse from the cell membrane down to every sarcomere. Because remember, the sarcomeres are the area from Z line to Z line. And there's probably billions of sarcomeres in every muscle cell. And every sarcomere is going to have a T tubule at each end. And there's going to be a sarcoplasmic reticulum surrounding every sarcomere. And they're going to have a terminal cistern at each end, packed full of calcium. So every sarcomere is surrounded by the sarcoplasmic reticulum. The sarcoplasmic reticulum runs the length of the myofibril, and which is running the length of the muscle. So this stuff is everywhere, packed full of calcium. The T tubule passes a nerve impulse from the surface of the cell down to the terminal cisterns of the sarcoplasmic reticulum. And the T tubules cause the sarcoplasmic reticulum, the cisterns, to open up and start releasing calcium and flooding the interior of the cell. We flood the cytoplasm, the sarcoplasm of the cell, with lots of calcium, an overabundance of calcium. We fill up this space with calcium, the very calcium that our bodies need for muscle contractions. This is where we need it for, why we need it. This is where we need it. Calcium comes out of the sarcoplasmic reticulum and floods the cytoplasm, the sarcoplasm of the cell. Because calcium is essential for contraction. Contraction is based on the formation of something called a cross bridge. The cross bridge is the connection between the actin and the myosin. When a myosin head grabs onto an actin, when the binding site on the actin is exposed and myosin grabs onto it, that's a cross bridge. That's the formation of a cross bridge. The actin and the myosin have to come together. And when they come together, the myosin then will pivot when it binds to the actin, it will pivot its head towards the middle of the A band, towards the middle of the dark band. And so whether they're on the left or the right-hand side, it doesn't matter. The myosin heads, once they grab onto the actin, will pivot towards the middle and pull on the actin, pulling the actin towards the middle of the sarcomere, and the entire sarcomere will shorten. And that's the point of contraction. And it will keep doing that as long as there's energy available from ATP. So normally our A band is made up of an overlap of the actin and the myosin. They're separated. When they're relaxed, there is no connection between the actin and the myosin. And there is overlap at the A band. But we have a model here that explains this. It's called the sliding filament model here that explains how thin filaments slide over the actin. I'm sorry, the thin filaments, the actin, slide over the myosin. Thin filaments slide over the thick filaments. They do not change their length. Their length doesn't change at all, but we slide thin over thick. When the nervous system tells us that we need to move a skeletal muscle, that will cause the thin filament, it will cause the myosin to grab onto the thin filament. The thick filament grabs the thin filament and pulls on it. That shortens the sarcomere. Every sarcomere shortens, and we get contraction of the cell. Because all the billions of sarcomeres in one cell all contract together with the same force at the same time. The cross bridges it only works if we have a cross bridge. This only works if the myosin head can grab onto the actin at the binding sites. That forms the cross bridge. Once the cross bridge forms, the myosin heads will pivot towards the middle of the A band. That drags the actin with it towards the middle. The actin slides over top of the myosin, and the entire sarcomere shortens. So when we pull the actins, we pull the Z lines closer together. The I bands get smaller. The Z lines get closer together. The A band gets thicker, gets darker. So what happens here? This is a sarcomere. I'll show you exactly what happens here. This is a sarcomere at rest. This is a Z line. This dark line over here underneath number 1 is a Z line. Over here at the end of the little tan box is the other Z line. So we have Z line to Z line. This is one sarcomere. Every sarcomere extends from Z line to Z line. This light area here is the I band. There's the dark band. There's the other I band. In the middle of the I band is the Z line. We have nothing but actin in here. And you can see there's the actin in here. And we have nothing but actin in place there. Over here at the A band, we have the overlap of actin and myosin. That makes it even darker. So we have thick and thin, and so on. So there's the A band, there's the I band, there's the Z line. There's an M line right here in the middle. Now, we're not worried about that right now. When we contract this sarcomere, it looks like this. The Z lines have moved in close. The I bands have gotten smaller. Because we're pulling on, the actins are sliding over top of the myosins, pulling the Z lines in closer. The entire sarcomere is shortened. The entire sarcomere has contracted. It has gotten shorter. And every sarcomere in the muscle cell is doing the same thing. Every sarcomere is shortening. Looks something like this. If you look at the animation here, you can see how the Z, when the sarcomere contracts, the Z lines move closer together. So our contracted sarcomere, Z lines are much closer than they were before. Let me go back one and take a look at this again. Now, your Z line at rest is way out here, but when contraction occurs, our Z line moves in here. We have tremendous overlap now because we're forming cross bridges between the actins and the myosins, and pulling on the actins, they slide over top of the myosin. The length of each filament doesn't change. The actins don't shorten, the myosins don't shorten, but the A band, and the A band doesn't shorten either. The A band doesn't change its shape either. The only band that changes is the I band. The I band shortens up as the Z lines are pulled in closer. And that's all. But that's how it works. We contract our muscle at the level of the sarcomere. Each sarcomere in a muscle cell will shorten at the point of contraction. The thick filaments grab onto the thin filaments and pull them towards the middle of the A band. A band doesn't shorten. The actin doesn't shorten. The myosin doesn't shorten. We're simply pulling on the Z lines, making the entire sarcomere shorten. As we see, the I bands come closer together, the I bands shorten, and the entire sarcomere shortens. And so we see, let me show you this one more time here. You slide over top of each other, and every sarcomere is doing it at exactly the same time with exactly the same force. So in the muscle cell, when a muscle cell is contracting, the billions of sarcomeres are all doing the same thing together. You know, if there's no, only if you get activated, everybody gets activated with the same strength at the same time. A muscle cell will contract to its fullest extent every time it's stimulated. There's no such thing as a partial nerve impulse. It's either all or nothing. It either is or it isn't. Once it's stimulated, it will contract. And it will contract to its fullest extent then. So. Now our brain controls all this. Our brain determines how we're going to do this. This is voluntary muscle activity. The central nervous system decides, okay, we're going to use a motor nerve here. We're going to contract certain muscle cells in our biceps to do something. And the term here that we've been using as a nerve impulse is really called what's known as an action potential. See, if you are an excitable cell, like a nerve or a muscle cell, you generate action potentials. Those are what we call nerve impulses. So the more properly termed an action potential, because you are changing the charge on the cell membrane of the nerve and the muscle cell. Every cell in our body has a charge, an electrical charge. Muscle cells run, the average charge for a muscle cell at rest is minus 90 millivolts. For a nerve cell, it's minus 70 millivolts. It's caused by the presence of ions inside the cell and outside the cell. So the action potential is a change in the charge on the membrane of a cell, of a muscle cell or a nerve cell. A nerve impulse, but we're going to call them action potentials right now. An excitable cell can generate an action potential. it can change its charge on its membrane. And it uses what we call a chemical called a neurotransmitter to do this. Now, because we'll find that the nerve never directly touches the muscle cell. The nerve never touches the muscle cell directly. It touches it indirectly. And so there is no direct connection. It's not hardwired. We use what's known as a neurotransmitter. The neurotransmitter is a chemical that allows the action potential from the nerve to be transferred to the muscle cell. It sort of allows it to jump from the nerve ending to the muscle cell and start again in the muscle cell. It's a built-in delay because we want to make, what it does is make sure that the muscle cell is ready. And the neurotransmitter we use in skeletal muscle is called acetylcholine. Acetylcholine is probably the most well-known and most well-studied neurotransmitter in our bodies. It's used in all skeletal muscle as a stimulatory neurotransmitter. There are about 60 different neurotransmitters in our body. Some are stimulating, some are inhibiting. So we'll see this when we get into chapter 11. We talk about the nervous system. So some neurotransmitters will stimulate, some neurotransmitters will inhibit. Acetylcholine in skeletal muscle is a stimulatory or an excitable neurotransmitter. So how does all this happen? Well, what I'm going to talk about now, the same thing we see with muscle cells also applies to nerve cells. Every cell in our bodies has a charge on it. And the charge is a result of the presence of ions outside the cell and inside the cell. And the ions in both muscle and nerve are going to be sodium and potassium. Now, we've got lots and lots of sodium outside our cells. Sodium is the #1 ion in plasma and in interstitial fluid outside the cells. Inside the cells, the #1 ion is potassium. So we have sodium outside and we have potassium inside. Now, the problem with sodium, the problem with potassium, as you probably remember, is that sodium will always leak in. High concentrations of sodium outside of a cell cause sodium to leak into the cell. And high concentrations of potassium inside the cell cause potassium to leak out. We don't want that. So we have to use the sodium-potassium pump to keep the concentrations, keep sodium high on the outside and potassium high on the inside. So, but the presence of these two ions, sodium outside, potassium inside, generate a charge, like a battery. So we tend to have a positive charge on the outside of the cell and a negative charge on the inside of a cell. I'll explain how a positive potassium can give us a negative charge. We use the term resting membrane potential because what we see is that with high sodium outside and high potassium inside, the cell tends to be positive on its outside and negative on its inside. There's more sodium there are more sodium ions outside than potassium ions inside. So you might have 3 or 4 times as many sodiums outside the cell than you do potassiums on the inside. So there are less positive charges inside the cell than outside. There's lots and lots of positive charges, lots and lots of positive sodiums outside the cell. There are fewer positive potassiums inside the cell. So even though they're both positively charged ions, if you have a lot of sodiums outside and fewer potassiums inside, the inside of the cell is considered negative with respect to the outside. In other words, the outside's positive and the inside's negative. And that positive outside and negative inside gives us a charge. We call it a resting membrane potential. This is what gives us the charge on the membrane. This is what gives us the minus 90 millivolts. Now, we're always pumping, sodium's always leaking in, and potassium's always leaking out. And we don't want that. We use the sodium potassium pump to pump sodiums out all the time and to bring the potassiums back in. Because if we were to reach equilibrium, the cell would have no charge and it would have no membrane potential, and it would not be able to do anything. It would be essentially dead. We always are trying to maintain the high sodium outside and the and we always want high sodium on the outside and low sodium on the inside. We want high potassium on the inside and low potassium on the outside. In other words, the outside of the cell is more positive than the inside. The outside of the cell is positive. The inside is negative with respect to the outside. Positive outside, negative inside. Sodium outside, potassium inside. And don't get hung up on trying to figure out why you have positive ions on the inside and it's still negative. There's just less positives inside than there are outside, so we're considered negative. The membrane potential is the difference between the charge from the outside and the inside. And the membrane potential on a muscle cell is about, a muscle cell at rest is about minus 90 millivolts. And our cells are polarized, just like a battery. Batteries have a positive end and a negative end. Well, cells have a positive outside and a negative inside. And that is the membrane potential. In muscles, and remember this, a muscle cell at rest has a membrane potential of minus 90 millivolts, a positive outside and a negative inside. And this is a great point to stop because I can't explain ion channels in 2 minutes. So we will pick up with ion channels on Thursday. Let's move along here. So let me get out of here. Any questions on any of this? You know, I know this has been, I'm trying, I don't want to confuse you with it. If you're confused, let me know. We'll talk about it in greater detail, you know, on Thursday. So anyway, I'm going to get us out of here. I got to get ready for my next class. And I thank you for being here. And I hope it wasn't too overwhelming. So I'll see you all. I'll see some of you in